SCUBE3 is a signaling protein produced within the dermal papilla, the small but powerful cluster of specialized cells located at the base of each hair follicle. The dermal papilla acts like a command center, sending biochemical instructions that determine whether a follicle grows, rests, shrinks, or regenerates. When SCUBE3 is released from these cells, it functions as a messenger that activates nearby stem cells in the hair follicle bulge region. These stem cells are responsible for initiating a new growth cycle. If they remain inactive, the follicle stays dormant. When properly stimulated, they begin producing a new hair shaft. Hair follicles operate in a repeating cycle consisting of the growth phase, known as anagen, the transitional phase called catagen, and the resting phase, known as telogen. In healthy follicles, this cycle continues for years. However, in thinning hair conditions, the anagen phase shortens while the telogen phase lengthens. Over time, the follicle miniaturizes. The hair shaft becomes thinner and weaker until eventually it may stop emerging from the scalp surface. SCUBE3 plays a role in signaling the transition back into the anagen phase by stimulating pathways involved in cell proliferation and differentiation. One of the primary pathways influenced by SCUBE3 is the TGF-beta signaling cascade. This pathway regulates tissue growth and cellular communication. In the context of hair follicles, TGF-beta activity must be balanced. Too much suppresses growth, but controlled activation in specific contexts can promote regeneration. SCUBE3 appears to interact with receptors that enhance regenerative signaling within dermal papilla cells, encouraging stem cells to re-enter the growth phase. This process involves activation of downstream transcription factors that promote follicular development. Dormant follicles are not necessarily dead follicles. In many cases, they are miniaturized but still biologically present beneath the scalp. The challenge is not replacing them, but reactivating them. SCUBE3 functions as one of the molecular triggers capable of sending that activation signal. When the dermal papilla is healthy and metabolically active, it secretes higher levels of growth-promoting proteins, including SCUBE3. If the dermal papilla becomes compromised due to poor circulation, inflammation, hormonal imbalance, or nutrient deficiency, its ability to secrete these signaling molecules declines. The dermal papilla is highly vascularized, meaning it depends on a rich blood supply. Oxygen and nutrient delivery are essential for maintaining cellular energy production within these cells. Mitochondria generate ATP, which powers protein synthesis and cellular signaling. If mitochondrial function is impaired, protein secretion declines. Supporting mitochondrial health indirectly supports SCUBE3 production because energy availability influences gene expression and protein synthesis capacity. Hormonal balance also influences dermal papilla activity. Androgens, such as DHT, can bind to receptors in susceptible follicles, leading to miniaturization. This does not directly eliminate SCUBE3, but it alters the microenvironment in a way that suppresses regenerative signaling. Over time, the dermal papilla shrinks, reducing its ability to communicate effectively with stem cells. Addressing hormonal imbalance can help restore a more favorable signaling environment. Inflammation interferes with signaling precision. Chronic inflammatory cytokines disrupt communication between cells. When inflammatory markers rise, oxidative stress increases. Reactive oxygen species can damage proteins, lipids, and DNA within follicle cells. This damage reduces their responsiveness to regenerative cues. Lowering inflammation allows signaling proteins like SCUBE3 to function more effectively because receptor sensitivity improves and cellular stress decreases. Mechanical tension within the scalp also influences dermal papilla signaling. Tight scalp tissue can restrict blood flow and create microinflammation. Reduced elasticity may compress follicles, limiting their capacity to expand during the growth phase. Mechanical stimulation, such as consistent scalp massage, may enhance circulation and stimulate dermal papilla cells. Physical stimulation can activate mechanotransduction pathways, where cells convert mechanical input into biochemical signals. This may complement SCUBE3 activity by increasing growth factor expression. Nutritional factors are foundational. Amino acids are required for protein synthesis, including signaling proteins. Deficiency in essential amino acids can limit production. Minerals like zinc act as cofactors for hundreds of enzymatic reactions, including those involved in DNA replication and cellular repair. Without adequate micronutrients, follicular cells cannot divide efficiently, even if growth signals are present. Vitamin D receptors are expressed in hair follicles, and deficiency is associated with impaired hair cycling. Adequate levels help regulate stem cell activation and differentiation. Metabolic health influences regenerative capacity. Insulin resistance impairs microvascular function and increases inflammation. Elevated blood glucose promotes glycation, which stiffens connective tissue and damages small blood vessels. This compromises nutrient delivery to the dermal papilla. Improved insulin sensitivity supports microcirculation and reduces oxidative stress, creating a more favorable environment for regenerative signals to operate. Sleep regulates growth hormone release. Growth hormone supports tissue repair and cellular turnover. During deep sleep, the body enters an anabolic state where regeneration processes accelerate. Chronic sleep deprivation reduces this restorative window, limiting follicular recovery and impairing signaling pathways. Stress elevates cortisol. Excess cortisol can prematurely shift follicles into the resting phase. It also increases inflammation and reduces immune regulation within the scalp environment. Long-term stress alters hormonal balance and suppresses regenerative signaling. The activation of dormant follicles through SCUBE3 signaling is not a single event, but a coordinated process requiring optimal cellular conditions. Dermal papilla cells must be metabolically active, adequately nourished, well oxygenated, and protected from chronic inflammation. Stem cells must retain responsiveness. Receptors must be functional. The extracellular matrix surrounding follicles must remain flexible enough to allow expansion. When these factors align, SCUBE3 acts as a messenger that encourages follicles to transition back into active growth. It does not override biological limitations, but it amplifies the body's intrinsic regenerative design. The focus, therefore, becomes supporting the internal environment so that this signaling protein can perform its function efficiently within the complex network of follicular regeneration pathways. Low inflammation and metabolic health form the biological foundation that determines whether hair follicles remain active or gradually decline. Hair follicles are among the most metabolically active structures in the body during the growth phase. They divide rapidly, synthesize large amounts of protein, and require a constant supply of oxygen and nutrients. If circulation is compromised, even slightly, follicles are one of the first tissues to show visible effect, TS, because they are not essential for survival. The body will always prioritize the brain, heart, and vital organs over hair production. Healthy circulation ensures that dermal papilla cells receive oxygen, glucose, amino acids, vitamins, and minerals. Oxygen drives mitochondrial energy production. Inside each follicle cell are mitochondria converting nutrients into ATP, the energy currency required for protein synthesis and cell division. Reduced blood flow lowers oxygen tension, limiting ATP production and slowing follicle activity. Over time, this can shorten the anagen growth phase and lengthen the resting phase. Microcirculation is particularly important. Large arteries may function normally while tiny capillaries supplying the scalp become restricted. Insulin resistance, smoking, chronic stress, and sedentary behavior all impair endothelial function, the inner lining of blood vessels. When endothelial cells become dysfunctional, nitric oxide production declines. Nitric oxide is essential for vasodilation, allowing blood vessels to widen and increase flow. Reduced nitric oxide means less blood reaching the follicle environment. Physical activity improves circulation systemically. Movement increases heart rate, enhances vascular elasticity, and improves nitric oxide availability. Even moderate exercise supports microvascular health. Mechanical scalp stimulation can temporarily increase localized blood flow. When circulation improves, nutrient delivery improves, and cellular waste products are cleared more efficiently. Inflammation silently interferes with this process. Chronic, low-grade inflammation alters the signaling environment around the follicle. Inflammatory cytokines, such as TNF-alpha and interleukins, can disrupt hair cycling by pushing follicles prematurely into the shedding phase. These inflammatory mediators also generate oxidative stress, producing reactive oxygen species that damage cell membranes and DNA. Oxidative damage impairs the dermal papilla's ability to communicate with stem cells effectively. Diet plays a central role in inflammation levels. High intake of refined carbohydrates causes repeated spikes in blood sugar and insulin. Elevated insulin promotes inflammatory signaling pathways and increases androgen activity in susceptible individuals. Excess omega-6 seed oils relative to omega-3 fatty acids shift the body toward a pro-inflammatory state. Highly processed foods often lack protective antioxidants that neutralize oxidative stress. Reducing inflammatory triggers through a nutrient-dense diet supports follicle health. Whole foods rich in antioxidants help protect cellular structures. Leafy greens provide magnesium, which supports vascular relaxation. Zinc contributes to immune balance and tissue repair. Vitamin C supports collagen production, maintaining the structural framework around hair follicles. Omega-3 fatty acids promote anti-inflammatory signaling and improve membrane fluidity. Metabolic health ties circulation and inflammation together. When blood sugar remains chronically elevated, glycation occurs. Glycation is the binding of sugar molecules to proteins, forming advanced glycation end products. These compounds stiffen connective tissue and damage small blood vessels. In the scalp, this can reduce elasticity and impair microvascular function. Poor glucose control also increases oxidative stress, compounding inflammatory damage. Insulin resistance reduces nutrient partitioning efficiency. Even if someone consumes adequate protein and micronutrients, impaired insulin signaling can prevent optimal cellular uptake. Hair follicles require consistent nutrient delivery to maintain rapid cell turnover. Improving insulin sensitivity enhances glucose utilization and stabilizes energy supply. Follicle cells. Intermittent fasting and carbohydrate moderation have been shown to improve insulin sensitivity in many individuals. When insulin levels decrease and stabilize, inflammatory markers often decline as well. Lower insulin reduces androgen conversion in certain tissues, which may indirectly benefit follicles prone to miniaturization. Balanced blood sugar supports steady energy production within dermal papilla cells. Visceral fat tissue acts as an endocrine organ, releasing inflammatory compounds. Excess abdominal fat increases systemic inflammation, affecting distant tissues, including the scalp. Weight management through diet and physical activity reduces this inflammatory burden. As systemic inflammation declines, the follicular microenvironment becomes more favorable for regeneration. Sleep also influences metabolic health. Inadequate sleep increases insulin resistance and elevates cortisol. Cortisol promotes glucose release into the bloodstream, raising blood sugar and contributing to inflammatory cycles. Chronic sleep deprivation, therefore, indirectly impairs circulation and follicle signaling. Stress compounds metabolic dysfunction. Elevated cortisol over long periods increases appetite for refined carbohydrates, disrupts blood sugar regulation, and promotes abdominal fat accumulation. Stress also constricts blood vessels, temporarily reducing scalp circulation. Over time, repeated stress responses create cumulative effects on vascular tone and inflammatory status. Hydration plays a smaller but still relevant role. Proper hydration maintains blood volume and supports efficient circulation. Dehydration thickens blood slightly, potentially reducing optimal microflow. Electrolyte balance supports vascular function and cellular communication. The extracellular matrix surrounding follicles relies on adequate collagen integrity. Collagen depends on vitamin C, amino acids like glycine and proline, and sufficient protein intake. If connective tissue stiffens Due to glycation or chronic inflammation, capillary exchange efficiency declines. Maintaining collagen quality supports both circulation and mechanical flexibility of the scalp. When circulation is optimized, inflammatory burden is minimized, and metabolic health is stable, the dermal papilla environment becomes supportive of regenerative signaling. Growth factors are produced efficiently. Receptors remain sensitive. Stem cells receive consistent oxygen and nutrients. Cellular waste is removed promptly. Mitochondria generate sufficient ATP to power protein synthesis. The hair cycle can proceed without premature interruption. Hair follicles do not operate independently from the rest of the body. They reflect systemic physiology. Improvements in vascular health, insulin sensitivity, inflammatory balance, and nutrient status influence follicular performance because they enhance the conditions required for cellular regeneration. Supporting circulation, reducing inflammation, and improving metabolic function create a biological environment where regenerative triggers can operate effectively within the natural hair growth cycle. Sleep, stress control, micronutrients, and scalp elasticity are often underestimated factors in hair recovery, yet they directly influence whether follicles remain productive or gradually weaken. Hair growth is not just a surface issue, it is a reflection of systemic repair capacity, hormonal balance, nutrient availability, and structural integrity of the scalp tissue. When these elements are optimized, follicles are far more likely to sustain long-term growth cycles. Sleep is one of the most powerful regenerative tools the body has. During deep sleep, growth hormone is released in higher amounts. Growth hormone supports tissue repair, protein synthesis, and cellular regeneration. Hair follicles in the anagen phase are rapidly dividing structures, requiring constant protein production and DNA replication. Without adequate sleep, the body shifts into a more catabolic state, prioritizing survival functions over regeneration. Chronic sleep restriction elevates cortisol and reduces insulin sensitivity, both of which interfere with the hair growth cycle. Melatonin, a hormone produced during darkness, also plays a role in hair biology. Beyond regulating sleep-wake cycles, melatonin has antioxidant properties. Oxidative stress damages follicle cells and disrupts signaling pathways responsible for maintaining the growth phase. Adequate melatonin production helps counteract this oxidative burden. Poor sleep habits, excessive nighttime light exposure, and irregular schedules reduce melatonin output, indirectly affecting follicle health. Stress management is equally critical. Acute stress triggers a temporary rise in cortisol and adrenaline. Chronic stress keeps cortisol elevated for extended periods. High cortisol shifts hair follicles prematurely into the telogen resting phase, contributing to increased shedding. This phenomenon is commonly observed after major life stressors. Cortisol also increases inflammation and disrupts immune balance around the follicle, potentially aggravating miniaturization in susceptible individuals. The nervous system has direct influence over the scalp environment. Chronic sympathetic dominance, often referred to as fight or flight mode, constricts blood vessels and reduces peripheral circulation. Over time, this can limit oxygen and nutrient delivery to dermal papilla cells. Techniques that activate the parasympathetic nervous system, such as controlled breathing, meditation, prayer, or Gentle stretching promote vascular relaxation and improve circulation to peripheral tissues, including the scalp. Micronutrients form the biochemical foundation of hair structure and follicle function. Hair shafts are primarily composed of keratin, a protein requiring adequate amino acids. However, protein alone is not sufficient. Enzymes responsible for keratin synthesis depend on minerals and vitamins as cofactors. Zinc supports DNA replication and cellular division. Iron is essential for oxygen transport through hemoglobin. Without sufficient iron, follicles may enter a resting state due to reduced oxygen delivery. Vitamin D receptors are present in hair follicles, particularly in the cells responsible for initiating new growth cycles. Deficiency has been associated with disrupted hair cycling. Magnesium supports hundreds of enzymatic reactions, including those involved in energy production and protein synthesis. Biotin contributes to keratin infrastructure, though true deficiency is less common than often assumed. B vitamins collectively support cellular metabolism and red blood cell formation, ensuring efficient nutrient transport. Selenium plays a role in antioxidant defense systems, helping neutralize oxidative stress. Vitamin A is necessary for cellular differentiation, but must be balanced, as both deficiency and excess can impair hair growth. Copper assists in collagen cross-linking and melanin production, contributing to hair strength and pigmentation. A balanced intake of micronutrients supports the complex enzymatic reactions required for follicle regeneration. Scalp elasticity is another overlooked factor. Over time, mechanical tension, inflammation, glycation, and poor circulation can cause scalp tissue to stiffen. The scalp contains connective tissue layers that must remain flexible to allow follicles to expand during the growth phase. Reduced elasticity may compress follicles and limit microcirculation. This mechanical restriction can impede nutrient exchange and signaling efficiency. Collagen integrity plays a central role in scalp flexibility. Collagen requires adequate vitamin C, glycine, proline, and sufficient protein intake for proper synthesis. Excess sugar intake accelerates glycation, stiffening collagen fibers and reducing tissue pliability. When connective tissue becomes rigid, capillary networks may function less efficiently. Maintaining collagen health supports both structural stability and circulation. Mechanical stimulation of the scalp may enhance elasticity. Regular, gentle massage can increase blood flow, improve lymphatic drainage, and stimulate mechanoreceptors in the skin. These mechanoreceptors convert physical pressure into biochemical signals that may influence growth factor expression. Improved tissue mobility allows follicles more room to function optimally. Hydration also contributes to tissue elasticity. Adequate water intake supports cellular hydration and maintains extracellular matrix flexibility. Dehydrated tissues are more prone to stiffness and reduced circulation. Electrolytes help regulate fluid balance within cells, ensuring proper communication and nutrient exchange. Hormonal balance intersects with sleep, stress, and micronutrient status. Chronic stress disrupts thyroid function in some individuals, and thyroid hormones are critical regulators of hair cycling. Low thyroid activity often presents with thinning hair. Adequate iodine, selenium, and overall metabolic health support, thyroid hormone production and conversion. When sleep is restorative, stress is controlled, micronutrients are sufficient, and scalp tissue remains flexible, the follicular environment becomes conducive to sustained growth. Dermal papilla cells can communicate effectively with stem cells. Growth factors are produced efficiently. Blood flow remains adequate. Oxidative stress is minimized. The extracellular matrix allows expansion during anagen. Hair follicles are better positioned to maintain longer growth phases and reduce premature shedding. Hair regeneration reflects a coordinated interplay between systemic physiology and local scalp conditions, addressing sleep quality, emotional stress, nutrient sufficiency, and mechanical scalp health supports the biological framework required for long-term follicle performance and resilience.